Perfect. I changed the title slightly. It's now called Replacing Eye Drops with Contact Lenses for Treating Cystinosis. So I think all of you are already familiar with the slides. I'm going to go through it pretty quickly. So cystinosis is a disease in which you get crystals in the eye, in addition to crystals in other parts of the body. And then you have treatments that can dissolve the crystals. And these treatments are typically delivered by eye drops. And these eye drops, as you know, are currently could be 8 times a day or 4 times a day. And that is a high frequency. And so that's the problem we're trying to solve. We're trying to reduce the frequency with which you have to deliver drugs to the eyes to treat cystinosis. So these are the drawbacks. And again, I think these have already been talked about in the talks today, and you're already familiar with it. Cysteamine, the drug that is used in these eye drops, which is also the drug we are using, is highly unstable in presence of oxygen. It reacts to form a drug called cysteamine, which is not active. And so that's this oxidation decreases the activity of the drug. And because of that, both of the formulations, the Cystaran as well as the Cysta drops, they have a short shelf life of just about 1 week, because after you open the bottle, the oxygen can get into the formulation, start reacting with the drug. And that's why after 1 week, really, there's not much of cysteamine left in the formulation. And again, for the same reason, Cystaran has to be shipped frozen, because oxygen can also diffuse into the bottle because the bottle is simply made of plastic. And so it has to be shipped frozen to minimize the possible oxygen going into the bottle and reacting. So these are the downsides or drawbacks of the therapy before you even instill the drug. So now I'm going to talk about what are the downsides after you instill the drug into the eye. So the biggest challenge with cysteamine eye drops, and really not just cysteamine eye drops, but eye drops of any drug, is that when you instill the drug, so here's the schematic. I'm going to try to use a pen to explain this point I'm trying to make. So imagine that you instill an eye drop into the eye. So what happens is once you put the eye drop into the eye, the drug is going to mix everywhere. The drug is going to go everywhere in the tears. And now what you really want is the drug that you're really interested in for dissolving the crystals, because the crystals are located here in the cornea, sorry, right here. This is where the crystals are. And so you want the drug to go into the cornea in this direction. But you can imagine that what will happen is some of the drug will also go into what is called, this is the back of the eyelid. This is the eyelid, this is the front, this is the back. So the drug will also go into the eyelid from the back side into a membrane called conjunctiva. Similarly, the drug will also go here. Now, this drug that goes into the conjunctiva is actually not wasted drug for cysteamine, because it turns out this will eventually go into the bloodstream, and which will be helpful in dissolving crystals in another part of the body. But nonetheless, the amount of drug that goes into the cornea is very small, because the area of cornea is only about 5% of the area of conjunctiva. Also, what happens is every time you're blinking, you're pushing some of this liquid out into the nose via an opening called puncta. So in about 5 minutes, what happens is all this drug is gone. So once you instill an eye drop into the eye, in about 5 minutes, all the drug is gone. And during that short time, only 1% of the drug actually, and I'll show you data for this, crosses across the cornea. And so that 1% is the only drug that really dissolves the crystals. The remaining 99% of the drug is not able to have any effect on the crystals in the eye. So only about 1 to 5% of the drug enters the cornea. And as I said, the majority of the drug drains out of the nasal cavity, or it goes into the conjunctiva. Now, because of this, because the small amount of drug is really permeating into the cornea, that is really the reason you need multiple eye drops every day. Another downside, which was mentioned earlier in one of the talks, is that these eye drops contain a preservative called benzoyl chloride. The preservative is necessary because if there's no preservative in the formulation, then bugs can enter the formulation and, of course, eventually grow and then eventually cause infection in the eye when you instill the formulation. So this preservative is needed. Now, the challenge with preservative is it's good inside the formulation, but it's bad in the eye, especially with multiple uses. There's a lot of data for the downside of using benzoyl chloride in glaucoma formulations. And glaucoma patients need to take it only once or twice a day. The cystinosis patients need to take it 8 to 10 times a day. So the downside of using benzoyl chloride is probably going to be even more, although there are not as many studies. And so because of some of these downsides, you have to deliver the drug 8 to 10 or 4 to 6 times a day, depending upon whether you use the viscous formulation or whether you use the non-viscous formulation. So these are the challenges we're trying to address with our approach. So we have done work in 2 different aspects of this disease. The first is, how do you improve the formulation? Because when we heard about this, that these drugs have to be shipped frozen and then they're usable only for 1 week, we thought there has to be a way to make them more stable, simply by changing the packaging. We're not changing the formulation, we're just changing the packaging. So I'll show you a little bit of data on that. And then the majority of the talk will be on how to use contact lenses for delivering drug for treating the disease. So first, let me just talk about the stability issue. So the reason why, there are two different reasons why the cystinosis formulations in these packagings, they don't last too long. The first has to do with some of the formulations. Now, I think one of the formulations is made in glass bottles, but the other one is in plastic. So if your formulation is in a plastic bottle, oxygen can actually diffuse across these plastic bottles. And so even before you open the bottle, what will happen is oxygen can get in, and then this oxygen get in will react with the cystine, and that will cause the degradation. So that's the first reason why the formulations can become destabilized. And that happens mainly in plastic bottles. In glass, this cannot happen because oxygen cannot diffuse across the glass, or at least not at the same rate anyways as plastic. The second thing that happens is that when you open the bottle, then every time you instill a drop, when you instill a drop, the drop goes out, but then the air comes back in, bringing oxygen with it. So that's the second reason why, that's the second mechanism through which oxygen can get in. And these two mechanisms together, essentially, that's the reason why, number one, you have to either make the formulation in glass bottles, or you have to ship it frozen to minimize this oxygen going in from the side. And the oxygen going from the top is the reason why after you open it, it basically cannot last more than 7 days. And so here is some data to show that. So this is what a normal plastic bottle formulation is, this data, that once you open it in about 500 hours, which is about 20 days, the entire formulation is basically completely oxidized into cystine. Now we can do something very simple. If we simply put a coating on the surface of the bottle that blocks oxygen, and there are plenty of materials available commercially, we can actually slow this down. So you can see by putting one layer, we can slow it down. So it lasts about 1,000 hours. And by putting 2 layers of these, we can actually slow it down considerably. Now, still it's deactivating. And now in this case, the amount of oxygen coming from the sides is basically almost 0, but still oxidizing because of oxygen coming from the top. So then we came up with an approach that we're not sure if really commercializable or not. At least it was a research project, but it may actually be commercializable. The idea here was that to prevent the exposure of this oxygen coming in, what we do is we put a layer of oily barrier at the top of the formulation. So this oxygen cannot go through the oily barrier, so then this drug is protected from even this oxygen. Now to instill the eye drop, when you flip the bottle, this oil actually goes to the top because it's lighter, and so you can still put the eye drop in. When we did that, you can see we can stabilize the formulation for at least a month easily. So this is some of the work, as I said, we did on the stabilization. The next, I'm going to talk about delivery. So to discuss the delivery, as I said, our path to increasing bioavailability. Bioavailability basically means what percentage of the drug that you put in your formulation is actually reaching the target. And as I told you, for eye drops, it's only about 1%. So our approach to increasing bioavailability is to load the drug in contact lenses. So let me explain why that approach is more effective, and then I'll also show you data to prove indeed it is more effective. So here's a contact lens, and we are going to put the drug inside the contact lens. So now the drug that is in the contact lens can either go out, diffuse out in this direction, or it can go out in this direction. Now the drug that diffuses out towards the cornea, most of that will actually go into the cornea because the drug can in fact diffuse out, get out like this, but because this distance is very long, the amount of drug that can get out from the side into the tear lake is very small. So almost the entire drug that gets out of the contact lens towards the cornea ends up going into the cornea, and it will cause the dissolution of the crystals. Now the drug that gets out from what is called, we call the front side of the tear film, this is called the pre-lens tear film, this is called the post-lens tear film, this drug will still not lead to dissolution, but nonetheless, 50% of the drug will actually lead to dissolution. So remember, the number is 1% for eye drop, and we expect this number to be about 50% for a contact lens. So as I said, we expect that 50% of the drug from a contact lens will actually make it into the eye. Other benefit is that the contact lens does not include any preservative, because preservative is needed really once you open the formulation to protect it, because when it's packaged, it's already safe because it was sterilized. And in our contact lenses are going to be single use. And so once you open the contact lens, you put it on the eye. So there's no need for the preservative. So you'll also address that problem. So here's the data, not for cysteamine. We are in the process of getting that data for cysteamine. This is for another drug called pirfenidone. We have this data for many drugs, but I included pirfenidone because it's a small molecule drug, hydrophilic drug, like cysteamine to some extent. So here we are measuring concentration in aqueous humor. Aqueous humor is the tissue behind the cornea. So there are two different data sets. One is the data with contact lens and one is the data with eye drop. So this is the data with contact lens, and you can see the concentration with the contact lens is significantly higher than concentration with eye drop. Now, this is even more amazing when you factor in that the amount of drug in an eye drop was actually more than that in a contact lens. In this particular study, the eye drop had 150 microgram of drug and the contact lens had only 60 microgram of drug. So the eye drops had more drug, but the concentrations are significantly lower. And once you do some calculations based on this concentration, you actually see that the contact lenses are about 50 times more effective compared to eye drops. Okay, so that was the data for a different drug. So now I'm going to, for the rest of the talk, I'm going to try to focus on, at least for the next few slides, I'm going to focus on cysteamine. So we started working on cysteamine. So to initially start with, we took some commercial contact lenses and we loaded cysteamine into the lenses and measured the release. So here we are plotting how much cysteamine gets released from a contact lens in vitro. In vitro means outside the body, you just do it in a beaker. And what you see is that the drug gets released very, very quickly. This is a percentage of drug release from 3 different lenses. And let's even look at the slowest release, which is actually with true eye. You can see in about 0.4 hour, which is half an hour, less than 15 minutes, the drug loaded in a contact lens is all released. Now, what that means is that if you use these commercial contact lenses for delivering cysteamine, you'll have to put a new contact lens every hour. And that obviously is not a very useful strategy. So what we want to do is we want to design a contact lens that can deliver the drug slowly. Instead of releasing it about half an hour, our target is about 4 to 6 hours. So it could be a daily disposable contact lens. So to do that, we came up with this invention where the idea to illustrate this invention, think of this as a contact lens. This is your contact lens, and the red molecules, think of this as the drug molecule. So if you put this contact lens in the eye, these drug molecules will essentially diffuse through the contact lens, travel through the contact lens, and get into the tear film. But now imagine that you create what we call diffusion barriers into the contact lens. If you create these barriers, then the drug will have to go around the barriers, and that will increase the time it takes for the drug to get into the tear film. To make this system very biocompatible, we designed barriers made out of vitamin E. Vitamin E is a very, very biocompatible molecule. There are many benefits of vitamin E, in addition to it being a good diffusion barrier. And we were able to successfully create the barriers in a contact lens. As you can see, this is what is called an SEM image. And you can see these are the barriers everywhere. And the lens remains completely transparent. The lens remains transparent because vitamin E is liquid. So the refractive index contrast is not significant. And also because these barriers are nano-sized. If you look at this scale bar is 4 micron, and you can see these are much smaller than the size scale bar. In fact, you can see it's about 50 nanometers in size. So very, very small barriers. And so the lens stays completely transparent. Now, if you look at the effect of this barrier incorporation on drug release, here again is cysteamine release in vitro. So the way we do this is we take our contact lens, incorporate vitamin E barriers into the lens, and then we load the drug and we measure the drug release. And here is the data, again, without the vitamin E barriers. You can see, I already showed you data. The release is about in half an hour or less. But with vitamin E, we can easily extend it to about 10 hours, 8 hours, 6 hours, depending upon how much vitamin E put in. And that's actually really nice because that means it gives us a knob that we can easily adjust depending upon how long, what is the duration that we want. So that's great. And after this, we wanted to measure, we wanted to make sure that, as Herbie was talking about, the first studies are always safety studies. Now, before we, of course, go to the humans, we need to go into animals. So we did safety testing of this in rabbits. We took these contact lenses, placed it on a rabbit eye for 24 hours. After 24 hours, we replaced the lens, put a new one in, and we did the study for 7 days. And after 7 days, we measured a bunch of different things just to make sure that the system, that these lenses have no toxicity. And at least in our 7-day study, we did not see any toxicity in the rabbits. After that, we wanted to do efficacy study. The challenge with efficacy study for cystinosis is with contact lenses is that both the mice and rat model that you have for cystinosis, unfortunately, they're too small for contact lenses. We can design contact lenses for these animals, but still the size is too small. There are challenges. We want to do these studies in rabbits, and there's a group in Michigan that is trying to develop rabbit models for cystinosis, but they're not there yet. So we did the next best thing. What we did is we took a cadaver cornea, and this is a cadaver cornea from a rabbit that is a normal rabbit, so there's no crystals. But then we managed to load it with crystals by loading it with cystine. So we took this eye, we put it in a diffusion in a cell. We filled this liquid up with cystine. Now cystine will diffuse in, and eventually it will oxidize to form cystine. So we did that, and actually it works pretty well. Eventually we managed to get crystals in the cornea. You can see the middle one. This is a confocal fluorescence microscope image from a human being with cystinosis. Other two images are actually images of our disease. And these images look remarkably similar. In fact, if you do a more detailed analysis, you can actually see that the shapes are pretty similar as well. So these are all needle-like. And that is something very interesting, that if you look at the shape of the cystine crystals in the cornea, they are needle-like, but they are polygonal in every other tissue. And I'll tell you in a second why that is. But nonetheless, you can see that the shape looks remarkably similar in our ex vivo model. This is the ex vivo model that we have created. This is the in vivo. And this is ex vivo, sorry. This is ex vivo model. You can see the crystals are needle-like. And then our hypothesis was that the reason why these crystals are needle-like in the eye is because of collagen. So we did some ex vivo studies without collagen, and we see that we get these polygonal shapes. With the presence of collagen, we get these needle-like. So what we have shown is that the reason they are needle-like in stroma is because of the collagen in stroma. Anyway, so the next thing we did was, after creating this model, we exposed it to contact lenses, and you can see we are very quickly able to completely dissolve away the crystals. So this suggests, this of course doesn't prove because it's a cadaver eye, it's not a real eye. This suggests that our contact lenses should be effective. And then we did some calculations to see equivalency of what is the equivalent amount of crystals we can dissolve with a single lens, and we get about 10 eye drops. So this suggests that our contact lens, we should be able to design a contact lens that will need to be only worn once a day for a few hours. If you need this contact lenses also have vision correction in them. So if you need vision correction, of course, you can wear it for the entire day. But if you don't need it for vision correction, you'll just wear it a zero powered lens, and you'll just have to wear it for about 3 to 4 hours and then just take it off. And that should have the same effect as the regular eye drop based therapy. And now we are also trying to create these crystals in live animals, and that's we have had some success, but we are not there yet. All right, so now that's where we are with cystinosis. But just to kind of show you that we in fact have tested these lenses in humans as well, not for cystinosis. I'm just going to share with you this data just to show you that the lens itself is safe, which means there's a very high likelihood that even the lens with cystinosis will be safe. So we did this clinical safety study in humans for glaucoma. So we made these lenses, and we put the lenses in 10 participants and for 2 days, and there was no safety issue. Then we also measured efficacy in glaucoma. Glaucoma is a disease in which the pressure inside the eye is high. So what we did was we took 2 subjects. It was a very small study. We're trying to recruit more patients. And we put the lens in one eye. So this is the lens in which we put the eye. We put in the eye. The other eye was controlled. And we measured the pressure inside the eye. So you can see as soon as we put the lens in, the pressure became low. And then it remained low for the entire duration of the lens sphere. And the control eye, the pressure also decreased because of what is called a crossover effect. And we got the same effect in subject 2 as well. So this shows that our lenses are effective, not yet in cystinosis because we haven't demonstrated that, but nonetheless in other diseases, they are effective at treating the disease, and they are also safe. So that's where we are. But recently we got some funding from Cystinosis Foundation UK and Cystinosis Ireland. Oh, sorry. And the purpose of this study is to measure the concentration in cysteamine in the eye with a contact lens. So the previous data I showed you was for pirfenidone, which showed that a lot of the drug can go into the eye. In this case, we're going to do the study with cysteamine. Now, this is still going to be normal rabbits. So unfortunately, we cannot show crystal dissolution. But what we're going to do is we're going to measure the concentration of cysteamine and from there calculate how much crystal dissolution can occur. In addition to that, we'll also show safety. All right, so to conclude, we think the cysteamine can be stabilized through oxygen-resistant materials. We think that contact lenses can deliver about 50% of the drug into the cornea, which can be used to dissolve crystals, which is significantly higher than with eye drops. So vitamin E loaded contact lenses, they showed safety in animals and also in ex vivo studies. So we think that that's at least the initial proof of concept. We also have done some human studies in glaucoma, which shows that these lenses are effective. So hopefully they'll be effective for cysteamine as well. And finally, this is not directly relevant to therapy, but nonetheless, the presence of extracellular crystals suggests more complex form. I didn't talk about this last conclusion, so I don't want to discuss that. But with that, I'd like to thank again Cystinosis Foundation UK for joining Cystinosis Ireland in funding our latest research. And I thank you all for your attention. I'll gladly answer some questions.